3-point mapping of genes in Drosophila melanogaster by Stephanie, Sarah, Shruti, and Holly. Thanks. 3-point mapping is used to construct a 3-factor map which shows gene-to-gene distances. In this experiment, we used Drosophila crossing a triple mutant and a wild type. The genotype of the wild type is B plus B, BG plus BG, BW plus BW, meaning light body color, smooth wings, and brick red eyes, respectively. The triple mutant has a genotype of BB, BG, BG, BW, BW, meaning a darker body color, wrinkled vestigial wings, and brown eyes, respectively. We can figure out the genotype of any of the flies we looked at, as the only possible combination for each gene are heterozygous and homozygous recessive, and distinguishing between the two is easily done simply by looking at the phenotype of the flies. Our hypothesis, can you go to the next one? Our hypothesis for this experiment was that the distances between the loci do not affect the accuracy of mapping data. To test this hypothesis, we needed to observe the phenotypes of Drosophila and construct a 3-point map. In this experiment, we looked at both male and female Drosophila after a wild type and triple mutant cross had been performed. We observed the phenotypes of many individuals, recording that data in a table. With all of this data from the cross, we were able to construct a 3-point map, which shows us the order of the genes and how far apart they are from each other. Okay.
In this experiment, the triple mutant Drosophila was mated with the wild type to produce offspring with clear phenotypes. The Drosophila were etherized and their phenotypes were recorded. The data was used to construct a 3-point cross with a map to map the distances between the eye color, wings, and the body color gene. Last, a chi-squared test was conducted to determine if there was a difference between the observed and expected distance values. The triple mutant Drosophila, comparing figures 1 and 2, it is clear that figure 1 is the triple mutant fly. Its visible darker body, red brick eyes, and vestigial wings are the classic phenotypes of a mutated fly. Figure 2 depicts a wild type fly because it has brighter red eyes, smooth wings, and a lighter body type. On figure 1, it is clear that the Drosophila is a male because there's a male sex cone present on its anterior leg. It is absent on the female's anterior leg in figure 2.
As we can see, is presented is the cumulative data of the cross between the mutant and the wild type. Each data point was collected from each group. The total number of flies was 475. Next. As we can see in the results, the calculated distance between the brown gene and the vestigial wing gene was 35.37 map units, and the calculated distance between the vestigial wing and black gene was 37.06 map units. This was calculated via using the 3-factor map. Next. Additionally, to view our results, we can see that the difference cannot be due to random chance due to the p-value being less than 0.05. Therefore, we reject the null hypothesis. There must be an external factor that caused the difference between in the observed and expected differences. Okay, next.
So the results showed that there was a significant difference between the expected and observed distances for the 3 genes as a whole, but just looking at the individual values, the observed and expected differences between the brown and vestigial genes are very close, and there was only a percent difference of 1.75%, but the difference between the observed and expected distances between the vestigial and black genes they were very different, and they had a percentage difference of 105.89%. So the original hypothesis is not supported by our data because the distances between loci does affect the accuracy of mapping data, and genes that are closer together, which in our case was the black and vestigial genes, were less accurately mapped. And since the observed distance between the vestigial and black genes was higher than the expected distance, there could have been more crossing over between the 2 genes, because when genes are farther apart, they have more crossing over between them, and that could have resulted in the phenotypes that we saw. So in the future, we would use a larger sample size and repeat the studies to see if we got the same results, and also larger sample sizes increase the accuracy of the data. And if repeated studies consistently find a significantly larger observed distance than the expected distance between vestigial and brown, we would have to design an experiment to figure out why this is happening. And this is interesting because you would normally maybe expect to see lower crossing over than there should be because of crossover interference, but our results showed that it was the other way around. And this could potentially be due to some environmental effects like UV rays and heat, but we would have to figure out why in future experiments. Thank you.